ready when you're ready. Okay, good evening, everyone. My name is Brian Gray. I am a professor of biology up at York College of Pennsylvania. For those of you in the Baltimore area, that's about an hour north along I-83. And my professional specialty, what I went to grad school for, is in the areas of medical microbiology and immunology. But I would like to talk to you this evening for, probably about half an hour about one of the things that I did my postdoctoral research on, which is actually looking at the ability of microbes to talk to each other. And they have very simple mechanisms for communicating with each other in many ways. And this comes out with an entire array of things that they can do. And then I'll provide some examples of some of the weirder interactions that microbes can engage in regarding sharing metabolisms, which is an interesting and funky topic. So my plan is for about the next half hour or so, I will natter on at you. I have a number of slides that I'm going to use, but then I would like to, if you have questions, go ahead and drop them in the Q&A, and I will answer them all as we go, and I'll open it up to a more, hopefully more active question and answer format for the last 15, 20 minutes or so. That's kind of my plan. And you know what they say about best laid plans. So I would like to share with you my presentation. So this is, of several topics I proposed, the Balticon, this is the one that kind of popped up and seemed the most interesting. And so what I'd like to start with is a brief survey of strange behaviors. Well, I wouldn't say strange for the microbes involved in them, what they can engage in, what they can do that actually requires microbes, bacteria in this case, talking to each other. And so a number of these photos, and I provided some of the links down here at the bottom, if you're curious about them. Excuse me. This is an example of bacteria that can produce slime. It's called a glycocalyx, and it's a protective measure. It prevents them from dehydrating. Here, what we see is a colony of bacteria that was dropped onto a plate and kind of just grew just a little bit right there. And here we see an example of an organism, a version, a strain. This is a strain that cannot communicate. This is a strain that can communicate. And what we observe is they scattered, they spread out across the plate. And that ability to talk depends on communication. And then we have two examples of microbial behavior tied into larger different organisms. One of them here, what you're looking at is a variety of seedlings. If I remember rightly, these are actually watercress seedlings. It's been a while since I've read the paper. And what you can see is they were laid out onto a plate of medium, and they grew roots. And so they grew roots and they were stretched out along the medium. But here, this was just the plants. Here, the plants were also sharing space with a microbe, a rhizobium, that actually influences and promotes root growth. And that mass of roots depends on the ability of the microbes to communicate with each other and, in fact, to communicate with the plants. And then finally, we have this terrifying organism. This is the Hawaiian bobtail squid. It is an absolutely massive 3 inches long. So yes, it's one of the cutest of the cephalopods. And what you might pick up on a little bit from this image is that there is some fluorescence here. This is actually a photo taken under natural sunlight, and that there is some ultraviolet light filtering through this water, and it's actually causing some fluorescence. But what you cannot see very well in this image is that there is a small space under the eyes of the squid that harbors, it's actually a specialized organ for these bobtail squid. It harbors an entire community of light-emitting bacteria. And what we have discovered is that at night, or actually at twilight, to be honest, when these small squid come out to feed, they spend the day hiding in the sand. At twilight, they will come out to feed, and these organs, these bacteria living in the organs, will emit light that will then mask the signature, the shape of the squid from below. It actually enables the squid to forage further out into the water, where normally deeper fish underneath them in the water column might look up and see this cute little short little meal snack above them. And the fact that the bacteria are talking to each other and this triggers them to emit light is protecting the squid. And in turn, it turns out the squid give a nice protection space for the bacteria to grow. So all of these processes, the making of slime, the motility, the promoting root growth, the producing light, they are all based on a process known as quorum sensing. So at its simplest, quorum sensing, you have a single microbial organism. I'm going to stick with bacteria. There is, for those of you aware of the domains of life, we have three. We have bacteria, we have archaea, and we have eukarya. We are eukaryotes. We are in the eukarya. And in general, every living thing that you can see with your naked eye pretty much falls into the eukarya category, the domain. Archaea are an interesting group of their own. They share some of the features of bacteria and eukarya, but I'm going to focus mostly on the bacteria this evening. So we have a small number of bacteria, or in this example, a single cell of bacteria, and it is producing a simple chemical signal. Now, when I give this particular slide with an actual live audience, what I'll start doing, it's a cute gimmick, I admit, but I'll stand at the front and as I'm talking, I'll start clapping. And this is the signal I'm emitting. Now, this is a signal, it's a small chemical being produced at a fairly steady rate. It's a low energy expenditure. As time goes on, this single cell divides, and there are now more cells in the same space, and they are in fact starting to produce the same quantity of signal per cell, and slowly this small chemical builds up. And in an audience, I nod to people in the front row and I'll say, please kind of start clapping. And so that is, you know, me as the microbe dividing and spreading and growing, and the signal keeps building. At some point in time, and this varies with the species, this varies with the signal, there is enough signal that what happens is that this crosses the threshold. And in fact, there are sensors on these microbes that will then recognize that signal and they'll respond to it. And so at this point in time, I'll say, okay, so I've got the entire room clapping, which, given that this slide is usually about 3 minutes into my opening presentation, it's a great thing to get applause that early into your presentation. But what happens when these cells receive this signal? They undergo a program change. They change their behavior, and now they are producing something else. My apologies. And so here in this little simplified diagram, we have a new material being produced. And so this is a program change for what we call transcription, the ability of the microbes to produce something and to respond. And so when I'm doing this in a live audience, what usually happens is I'll say, all right, what's the program change? Everybody please stand up. And I thank them for giving me a standing ovation so early in the audience. It's a great shtick. But that's the basic idea. Produce a signal, grow, make more signal. The signal accumulates. At some point, the signal is received by the same organism, and then a program changes. And there are a number of different examples of this. Let me give you some examples of that. So Vibrio fischeri is the organism that emits light in the bobtail squid. And what we have there is a fairly straightforward circuit. It's called the Lux Circuit, or for those of you who know your Latin, light. And we have one enzyme. So this entire big oval is the bacterium Vibrio fischeri. It is producing these enzymes, these genes. We have Lux I right here, and it is producing this small chemical represented as a pentagon. For those of you who are curious, it's something known as a lactone, which is actually a ring structure. And it secretes these small chemicals out into the environment around the bacterium. The concentration of this molecule builds up, and at some point in time, since the chemical will pass back and forth the membrane, there is another protein, a receptor, that is named, simply enough, Lux R. And when Lux R binds to this signal, that changes Lux R, and it instead moves to the DNA of the bacterium and initiates new programs, new activities. In this case, Lux R will bind to the DNA and start the production of luciferase. Some of you might be aware that luciferase is the name that we have given to enzymes that will produce light from chemical reactions. Yes, there's a play in there on Lucifer the Lightbringer. But these are basically the same sorts of proteins that we see in fireflies. And fireflies have their own version of luciferase. Completely different mechanism for turning on light production, but we gave the light-producing enzymes the same name. Go figure. So this is the very basic circuit for producing light in Vibrio fischeri. Produce an inducer. As the inducer gets to a high enough concentration, as the signal gets loud enough, that signal is heard by a receptor, and this initiates a new program. Sounds kind of like basic computation in some way. Now, for my own graduate work, what I was involved in was pathogenesis, the ability to cause disease for this particular pest. Many of you may have heard of organisms known as MRSA or VRSA, very potent antibiotic-resistant versions of Staphylococcus aureus. Now, Staph aureus is an organism that many of us have naturally and normally inside of our sinuses. It's actually up here at if you were to put your fingers on your cheek, right where your cheek bends into the base of your nose, that's roughly where Staph aureus is in about 60% of the human population. And if it stays in your nose, Staph aureus is just fine. It doesn't actually do anything else. It's actually kind of benevolent if it stays in your nose. It's when Staph aureus gets into other tissues that it becomes far more of a problem. It turns out that a large number of strains of Staph aureus also engage in quorum sensing. They engage in this ability to recognize that the number of Staph aureus bacteria has reached a certain threshold. And so that's really what you're seeing right here. Across the bottom is an example of a particular organization, of genes in bacteria. And bacteria, because they're so small, are very efficient in many ways. And one of the things that they do is that they will organize related genes in a very tight, compact little cluster. And they will produce all of them at once if they're involved in a single function. And so here is the genetic marking for this particular group. And as you can see, they correlate with various components of this system. And so it's a slightly different system than what Vibrio fischeri uses. Here we have the D protein. In this case, it's a protein. It's not an organic chemical like the lactone from Vibrio fischeri. This protein, AGRB, will chop up and actually fold the D protein into a very small molecule called AIP. This stands for auto-inducing peptide. There's that terminology again, the induction. And that's what they're doing. And so B will grab this protein from inside the cell, chop it up, twist it up, and then spit it out into the environment outside of the cell. And so this is out, it's been secreted. It's been dumped out into the environment. Now, if we're talking a disease process, an infection, this might be in a tissue wound. This might be on a needle stick. This might be in the urinary tract. It depends entirely on where staph wound up that it shouldn't be. The concentration of this small molecule, it is cheap to produce, so it's easy to make it and throw it away. That's something in common about all of these small molecules, is that it's not a huge energy expenditure. And so they can get tossed outside of the cell body and basically disposed of if that's the way the environment works out. The concentration of this maybe will build up, and if it hits a certain threshold, it's recognized by this protein, this enzyme. The receptor here is the C protein of this cluster of genes. And this is an enzyme that will activate another protein, AGRA. And AGRA will, in this case, the first thing it binds to is the gene set for the AGR operon. So in Staphylococcus aureus, this entire system is a positive feedback loop. If we're looking at the Vibrio fischeri, back up one, this is not a positive feedback loop. This is, here's an inducer, it's being made at a steady rate, and it at some point crosses that threshold and light gets produced. That's a very simple on-off system. This, in Staph aureus, massively ramps up the production of this inducer molecule and then activates and turns on even more of this system right here. So it's a positive feedback loop in staph, which for those of you who have had any experience in understanding signals or processing or so on, it gets very messy very quickly. Other things that AGRA can bind to and activate, it turns out, are pretty much the entire pathogenic disease-causing protein set in Staph aureus. And so what was observed in skin infections, and this is where my research was looking at skin and soft tissue infections within Staph aureus, was that when these bacteria are trapped in a patch in the skin, say a puncture wound or a laceration, as the bacteria grow in this itty bitty space and they start producing this signal, what they're doing is they're using up all of the easily consumed nutritional resources. So they're using up anything that might have come in with them. They're chewing up anything that's easily digestible. And then what'll happen is at a certain point, as this signal accumulates, as there are enough bacteria, then this new program is turned on. And this new program involves the production of an entire series of digestive enzymes. And those digestive enzymes will chew up our proteins, our fats, they'll attack our cells, they'll attack our immune system. And basically, what is turned on in this new program for Staphylococcus aureus is a way for the bacterium to get more food. Now, those of you who might have caught my talk last night, or my panel last night with Dr. Caitlin Ahrens, I had talked about how bacteria really are only looking to get food and get nutrients in many ways. And so what we are as giant self-important thinking organisms We like to think we're the most important thing in the world in many ways. And bacteria are kind of like, yeah, you're just a big old pile of food. You're a big mobile pile of food. Gimme, 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 gimme. And so all of our hard-won efforts in all our cells built together and so on to bacteria, they're just special piles of food. So this is kind of the dark side of the quorum sensing. This is quorum sensing processes involved in disease. Now, back to this set of examples right here, looking at examples of quorum sensing that do perhaps more positive things. Here, and with both these seedlings and with the bobtail squid, we have examples where there is an interaction between the bacteria when they talk to themselves and the host organism that maybe makes some space for them. So what we have with these seedlings, some of you may have heard the term the rhizome, which is in some plants, it's actually a special sort of coating around the roots of the plants. And we have found that some plants, for example, legumes, these require things called nitrogen fixing bacteria to make use of, provide nitrogen from the soil. And so the legumes provide the rhizome as a home for these bacteria. And the bacteria that will grow into the rhizome, they'll move into that little space. They will not start cranking up their nitrogen fixation machinery until there are enough bacteria of the same group present in one space. So a physically constrained little space. Then those nitrogen fixing bacteria, when they are dense enough, will start producing those enzymes. They'll start fixing the nitrogen and they will provide a benefit to the plant. So it's a give me some room to grow and I'll pay you back. And so this, in the case of plants, sets up a lifelong interrelationship between the bacteria and the plants. If we're talking about the Hawaiian bobtail squid, it gets very interesting in that regard. The particular organ that is just under the eyes of the bobtail squid is open to the outside environment. And what we have discovered is that every morning as the sun rises, these squid will actually pump like little bellows. They will flush out these little organs under their eyes, and they will squeeze out all of the bacteria that have been emitting light all night long. And then they will take in fresh seawater and basically rinse out these small spaces. That sounds rude. It sounds like you just kept me alive and now I'm done with you. Well, it turns out that the squid have specific structures and specific molecules on the surfaces of their skin inside these little organs that only allow Vibrio fischeri to grow. And so it is a guaranteed safe place for the bacteria to grow. But why dump them back out in the environment? Why spit out all of these bacteria that just kept the squid alive all night long, presumably? Well, it turns out that the squid hide during the day, and they don't need the bacteria to produce light. And it's a very interesting relationship. First thing in the morning, the squid flush out those organs, and that leaves just enough of the Vibrio bacteria that those bacteria will grow at a rate that they will hit their quorum sensing threshold at sundown. And so as the sun sets, there are now enough bacteria that they will turn on their light producing ability and they will start to luminesce at the end of the day. So this is an utterly fascinating interplay and relationship between the bacteria and its host, the squid. that there's this interplay of timing, there's this dependency on the rate of growth in many cases, back and forth, which as a biologist, I find utterly fascinating. If you're curious about this one especially, I recommend the work of Bonnie Bassler. She is a professor of biology at Princeton, and she has done a great deal of work on this particular system here. Now, some of you might have been listening along and thinking about the potential benefits for what we now call synthetic biology or bioengineering. Can we turn, since we're clever tool-using primates, can we turn the discovery of these biological systems into a more active and engineered process? And the answer appears to be pretty clearly yes. So this is actually from a recent review. My bad, my apologies. I didn't save the reference here. But this is a review of synthetic biology using quorum sensing systems. And the basic idea is, well, since we have genetically defined discrete quorum sensing packages, can we drop those into other types of bacteria? Can we enable quorum sensing in other groups of bacteria, maybe ones that do other things? And can we combine these possibilities in various different ways? Can we cut the system in half, that one group of bacteria make the induction molecule and the other group of bacteria make the have the recipient? You know, what are possibilities in there? What can we do with all of that? And the answer appears to be yes. This is all made possible through genetic engineering, all the modifications in there. And so here we have an example where we could, for possibilities that are currently beyond me, I'm not trained in computational science, but I understand the idea of simple logic gates. So you could do some sort of simple biological programming based on who's making the signal and who's receiving it. We can have crosstalk happening where different species are making an inducer and different species are making the receptors. And there is some evidence this actually occurs naturally, that prey species make an inducer molecule for their own benefit, but we're starting to see predatory bacteria, or I should say, we're starting to find them. They've probably always been there, but we didn't know to look for it. Predatory bacteria that recognize that signal, and then they will home in on the prey species. We can have multi-stage biological processing, chemical synthesis, but these days we require multi-stage, chemical production processes where, okay, we make this first chemical modification, we have to purify the product of that chemical reaction so we can do safely and with high efficiency the next process. Here, though, we start with the input. One bacterial species produces the intermediate, a second species can process it to the final product, and as this organism grows up, it sends a signal to the next one in line saying, start your machinery, do the processing. And various other aspects in there. These are all different possible examples for what can be done with this, just the simple idea of quorum sensing in many ways. So I'm gonna stop that for the moment, and I will actually open up questions if people would like for the Q&A. And I've got one more slide if people want to listen to that. But if you're curious about any things, please let me know. Or else my nice mellow baritone has stunned you all into postprandial sleep. How complex can the computations be? That's a good question. So I'm seeing this from SB. How complex do you want them to be? We find in living examples of, so I'm providing a very simplified version. We are aware of a number of species of bacteria where they are capable of depending on which strain of bacteria you're looking at, 16 or 17 different molecules that they all have the same basic structure, but the strain variations mean that they have a plethora of inducers and a plethora of receivers. So when you've got that kind of fine gradation between the ability to send a signal and ability to receive it, and because of molecular technologies, we can slice and dice and mix and match. I mean, computers seem kind of simple with ones and zeros. I understand qubits will get a little more interesting, but if you've got the possibility of basically hexadecimal basic computing inside living systems, how fun is that? So that was SB's question. I'm guessing John Seal was agreeing with the question or the answers therein. So other questions? Ooh, Jerry asks a good question. Does this bacterial communication work with fungi like the mycorrhizal fungi? Ooh, so I think this is one of those situations where we haven't looked yet. I'm guessing the answer is very strongly yes. It's just a matter of nobody has yet hasn't found enough evidence, but that may be because they haven't looked yet. So one of the things that I did not mention up front, because this field is expanding very rapidly, is that there is a huge growing body of evidence that shows that bacteria do in fact communicate with us, and that bacteria also communicate with plants. So when I mentioned the rhizome, there is some evidence that bacterial nutrient processing will actually influence the growth rates of the plants that host them, and can furthermore influence the size of, the size of leaves and the particular pigments that are being produced in those leaves. So a darker green or a lighter green. What we're finding in our bacteria, so this is a lot of work going into the gut microbiome, the concept of all of our microbial passengers that live in our intestines. So depending on which bacteria we have present in our intestines and which particular mixture of things they're producing, our neuroendocrine systems will recognize and respond with things like satiety, with hunger, with depression, with anxiety, with suppression of anxiety, with mood elevation. So the microbes in our guts, it's unclear at this point in time how much we're signaling back to the bacteria. Certainly with what we eat, we can influence how the bacteria can respond to us. And so you are probably going to see in the next decade custom tailored, here, take this pill of bacteria. It's going to influence what's growing in your gut. You're going to take, this entire series of pills for a month, and that will influence type 2 diabetes. It'll influence depression. It'll influence your ability to respond to chemotherapy for certain kinds of cancer. These are all studies that are going on that we're finding about the influence of the gut microbiome on the host organisms, us. Danny asked a question. These are slime molds, right? So slime molds are simply one of the groups of bacteria. By the way, for those of you unaware, slime mold is a huge misnomer. Dictyostelium, which makes what are called slime molds, are in fact a form of bacteria. It's just that they cluster in large groups that we think are more complex organisms. So no, Dictyostelium, slime molds, I love the fact that the closed caption gets that right. I'm really impressed. Anyway, sorry. I love technology. So the slime molds the dictyostelium, are having multiple bacteria communicate and cooperate. And they appear to have several different subsequent cascading signals after the first signal has induced the first new behavior. One of the things that gets turned on is the second signal gets produced. And this, as it builds up, if there's enough sufficient mass of bacteria, will initiate the second behavior. And so we'll get the pigment production, we'll get the apparent movement of the entire slime mold, and the branching behaviors of the slime mold. This will in turn start production of the third signal, which will initiate a third behavior, and so on, cascading sequence of responses. So slime molds are simply one of the groups of bacteria that can do this. SB asks again, can different strains of bacteria communicate, like different colonies saying hello? This is a very good question because it's an interesting problem where the entire complexity of the signal appears to be, hi, I'm here. Hi, I'm here. Hi, I'm here. And that is the entirety of the signal. There doesn't seem to be an ability to have conversation with information any deeper than the, oh, there are enough of us here now that changing programs and doing something new is good. So that the entire density of information is how many of us are here in one spot. Now, there is some evidence that the bacteria, there is evidence of cross-species communication. And that is being researched, because the only time you ever find a pure culture of a single species of an organism is in a laboratory. Every other naturally occurring presentation of bacteria, except for weird things like Vibrio fischeri in bobtail squid, every other time we see bacteria in nature, they are in a multi-species community. So yeah, there's evidence that bacteria, multiple species, if they're sending and receiving the same signal, they can basically say, yeah, hi, I'm here. And when that number gets high enough, things happen. But we haven't investigated that yet. I keep trying to, but that'll take me, if I had about $5 million to buy the equipment, I'd be really, really happy to try. That's a tough one, but it's a good one. Other questions. Ooh, good ones. Okay, so let me jump down to Clint. Do we have a system in our noses to clear out the inducing peptide from Staph aureus? Yes, actually. And I love talking about, it's a particular paper that I introduce my students to when I talk about the concept of the bacteria we have on us and things like physiology, mucous membranes, like mucous membranes in our noses, in our eyes. There is a research group out of, I want to say it was Germany. What they did was that they sprayed with an aerosol a fluorescent dye in a very narrow band right here in the nose. And they timed it. They timed their test subjects and said, okay, do we actually see mucus move in somebody who's not congested, having runny nose from allergies, et cetera, et cetera? And the flow rate of mucus from the front of your nose to the back of your soft palate is about 2 centimeters an hour. I love that research because it's one of those, that's like classic Ig Nobel Award territory. It's kind of like, how did we wind up doing that? And so, yeah, the natural production and flow of mucus will actually drain all of this inducing signal from where in our sinuses Staph aureus grows and keeps flushing it down the back of our throat into our stomachs, where stomach acid digests it. And so, yeah, we have a system in our noses to clear out the inducing signal, the AIP from Staph aureus, and it also keeps the numbers of Staph aureus actually, generally speaking, down below that threshold. And we have that on all of our mucous membranes, in our GI tract, gastrointestinal tract, in our genitourinary tract. We have this mucus production that has this kind of constant flushing going on. Is the management of signal sending gut bacteria under investigation? Yes, absolutely. It's one of the hot areas of research right now. Thanks for that question, John. There are many labs across the globe that are exploring, okay, what is the extent of communication, not just from the bacteria to us, but from us back to the bacteria. It is something far below our conscious level so far. But yeah, no, we're absolutely exploring, can we influence the behavior of our guts? Can we alter how the bacteria are digesting? Can we alter that? And what species do we need to have in our guts to improve our health? Extremely active area of investigation in many ways. Yeah. Danny asks, can you trigger a behavior shift prematurely by artificially adding the signal molecule? Yes, absolutely. There was a fascinating study that came out, 2012, where a bunch of microbiologists got together with a bunch of engineers, and they said, oh, you specialize in making microcapsules. Can we work together? Can we, using your technology, encapsulate single bacterial cell inside this space, inside a space that's only big enough for that one cell? So we're forcing it can't divide. And so it turns out that if you have just a single cell, it can, in about 20 minutes, make enough signal to turn itself on. So you've got quorum sensing where the quorum is 1. And so, but yeah, no, we actually, in my research, we bought synthetic AIP for Staph aureus, and I would add that to cultures to start processes early. So we can absolutely add artificial signals to trigger this process very, very early in the bacterial span. No, absolutely. Okay, getting back to SB. Given how much bacteria are beneficial to plants and animals, including us, how harmful can antibiotics be to the ecosystems? They're terrible, honestly. Some of you may be aware that there is an initiative to reduce antibiotic usage across basically all of medical practice in many ways, because of the damage that antibiotics do to our microbiomes in many ways. And one of the problems that really kind of drove this home is recognition of what's called antibiotic-associated diarrhea. So this is a problem seen in patients who are taking multiple antibiotics. So somebody who has one doctor dealing with bronchitis, another doctor that they see dealing with a skin infection, another doctor they see dealing with a urinary tract infection. And so each of these get a different antibiotic, and they wind up, because the doctors originally weren't talking to each other. This would totally wipe out the gut microflora, basically blitz and steamroll their gut bacteria. And this opens literally the door, or gives space for Clostridium difficile to grow in that space. And so, yeah, C. diff, and I see a comment actually from Hobbit that gut flora transplants, nicknamed poop soup. Yes, that is actually the name the labs have given to it, poop soup. Transplants of gut bacteria from healthy patients to patients with gut disease to recover from and restore from the overuse of antibiotics in patients with antibiotic-associated diarrhea. Yeah, so we are cutting back on our antibiotic usage in patients. Antibiotic diffusion out into broader spaces is not as much of an issue because it dilutes. It dilutes in water hugely. It'll dilute through soil and so on. And so The only reason really we see it happening in people is because we are giving a high dose of antibiotics in a confined system, fundamentally. Is there a feedback loop that tells mass-produced immune cells that after defeating? So Jerry, you're asking a question verging into my other specialty, immunology. So it depends entirely on which immune cells you're talking about. And I'm gonna save them, and I'll actually pop over to the Discord after this if you're curious about having, continuing on that one, because that's an entirely immunology-based question. So I'll get to that one there. And then John asked a question: what is the quanta of bacterial information? Well, it depends on the species. It depends on the system. So some receptor systems have a much lower threshold of induction signal that they'll respond to. Others have a really, really high threshold. And so it depends entirely on the system. And some of them are very insensitive. Some of them are very sensitive. And this, I haven't seen a good analysis yet of correlating the sensitivity with the program that is then induced and the program that is then affected. We haven't gotten there yet. I know there are synthetic biologists who are exploring that. So that's a good question. It's a deep question, one we haven't actually answered yet in that regard. So I think we're getting there. Okay. Dilution is the solution to pollution. Yes, absolutely. I opened up the chat. Yeah. So my understanding is we've got about a minute left by my clock. Are there any other microbiology questions people are curious about in this topic, in this area? Oh, I've got 3 minutes left. Okay. In that case, then I'll take advantage of. What about antibiotic resistance? Danny, what about it in particular are you curious about? Because that's a really big, broad question in microbiology. Ooh. So is antibiotic resistance something that can be triggered by quorum sensing? Honestly, we haven't seen that. Because most bacteria that are antibiotic resistant, it is a feature that is basically always turned on. Now, we can certainly engineer a system, that will turn on antibiotic resistance in response to cell signal. But in nature, if an organism is antibiotic resistant, that resistance is always expressed as natural occurrence. And so for those of you who may be unaware, all natural, so if the term is antibiotic, it means that it is actually produced by another microbe. And so what we've done with penicillin and spectromycin and bacitracin and so on is we have taken advantage of bacterial and fungal chemical warfare. They bump each other off. They actually hate the competition. And so for every naturally developed antibiotic, so anything that's a first generation antibiotic, we are going to find natural resistance to, because the bacteria have been making it, and the fungi have been making it, and they have naturally evolved resistance to it. So yeah, it's not something that we see in, we don't see it inducible in the wild. We see it, but we can make it inducible in the lab. Ooh, Jane asks a fantastic question. All right, the flip answer, Jane, is how much money do you want to throw at the problem? At what point will scientists know enough to create a tree showing all the various types and levels of sophistication for communication between them? Ooh, I haven't seen one yet. We've been aware of quorum sensing since the late 1980s, and we still have only figured out gene information for maybe 7% of all the bacteria out there. So even though sequencing technology for understanding the gene and annotation technology has developed tremendously in the last 15 years, I'm not expecting to see that for another 20 years or so, because we're going to have to be able to sequence out genes from thousands upon thousands of species. And there are probably several million different species of bacteria out there. And so I would honestly love to see a tree or a map like that, Jane, but it's not going to be anything that we're going to see anytime in the near future until we have a greater handle on more different species of bacteria out there and all the different systems they use. So I'd love to see that, but it's not ready yet. So. All right. Okay, going to the Homeland channel. All right, I will pull that up. Anybody wants to hit me up with some more questions over there, I will be able to talk to you shortly. I'm there as Dr. Brian Gray, by the way. Okay, and Jerry, I will, if you pop over to the Discord, if you can, I'll answer your question about immune system apoptosis over there. So. You're very welcome, everyone. I hope it's been informative, interesting, and given a bunch of you some interesting ideas to play with. All righty.
Oh, that was a lot of fun.
Thank you much, Hobbit. Yeah, like I said, it's a lot of deep, interesting stuff in there, and there's a lot of room to play. So
we were investigating this stuff with my mom about 1.5 years ago. It turned out she was lactose intolerant, but she's having all these gut problems, and we were discussing fecal injection and all that. And I was like, okay, yep. Eat shit and not die.
Yeah, although I believe the preferred mode of application is actually a bacterial enema.
So it goes in either end, but it goes way in without.
Yeah, it has to go to the large intestine, has to go to the colon.
The initial processing.
Yep.
It was still funny to think about.
It is, and the fact that the labs call it the poop soup, it doesn't help.
That's awesome.
All right, Hobbit, thank you very much. I'm going to go pop over to Discord and be well.
Yeah, you too. See you on the next one.